Hi, my name is Alana Marte, and I will be presenting my cell membrane model and explaining it. I will first be defining some of the cell membrane structures. First up, we have phospholipids, and as you may guess from its name, it's a type of lipid that has a hydrophilic head and hydrophobic tail. Next, I have the integral protein, which is embedded in the plasma membrane and has a range of important functions, such as channeling or transporting molecules across the membrane. Next are the peripheral proteins, which form temporary bonds with the cell membrane, allowing them to detach and reattach at specific times with specific signals. Next is the cholesterol, which has a role in the membrane fluidity, but its most important function is reducing the permeability of the cell membrane. Keep in mind, cholesterol is only present in animal cells. Next is glycolipids, which are generally found on the extracellular face of eukaryotic cellular membranes and function to maintain stability of the membrane. Next is glycoprotein, which, like glycolipids, are found on the extracellular face of eukaryotic cells, and they act in cell-to-cell recognition and binding of other molecules. And lastly, next is the cytoskeleton, which is a network of protein fibers that run throughout the cytoplasm, where it is responsible for support, mobility, and regulation in some biochemical activities. This structure illustrated here is of an animal cell's plasma membrane. The extracellular side is the outside of the cell, and the intracellular side is the inside of the cell. The phospholipid tails attract each other, which result in the phospholipid bilayer. In the middle of the phospholipid bilayer is the cholesterol, which is attracted to the tails of the phospholipids. The integral protein is on the left side, the glycolipid is on the surface of the cell on the right side, and the glycoprotein is on the surface of the left side. The pink and green circles on the inside of the cell represent the peripheral proteins, and the microfilaments of the cytoskeleton are shown inside the cell too. The two nucleophiles in the middle represent proteins, which substances go through, which will be demonstrated later in the video. In this illustration, it is showing simple diffusion. Simple diffusion is basically the passive transport of substances without the aid of membrane proteins. So, the substances are passing through the phospholipid bilayer without the help of protein channels or ATP. As we can see, the gradient orientation is from high concentration on the extracellular side to low concentration on the intracellular side. An example of this is when oxygen moves into the cell along with the gradient. In this illustration, it is showing facilitated diffusion using two different types of transports. The first type of transport is shown through the channel protein, where the substance goes straight down the protein without stopping. The second kind of transport is through the carrier protein, which stops the substance before letting it pass through. In both these types of transport, no ATP is needed, and its gradient orientation is from high to low concentration. The example of the substance going through channel protein are ions and for carrier protons, sugar. This next illustration is showing active transport, which needs ATP. The example is showing active transport using the sodium-potassium pump. As we can see, when 3 sodiums from the intracellular side go through the pump, 2 potassiums from the extracellular side go inside the cell. The gradient orientation for this pump is going from low to high. The next type of active transport I have illustrated here is the proton pump, which demonstrates one hydrogen moving out of the cell from low to high concentration. The last type of active transport I chose to include is the cotransport of sucrose, which is sugar. Since sugar is too big to go inside the plasma membrane by itself, it needs to be accompanied by a molecule, like sodium molecule, for example. In my example, we can see a sugar molecule going into the cell with a sodium molecule. When this is happening, a sodium-potassium pump is happening simultaneously, but I have separated here. In this illustration, it is showing endocytosis, which is how large molecules enter a cell. In my example, it shows how the cell changes its shape to surround the molecule, allowing it to enter the cell. An example of this is when water enters the cell. In this last illustration, it demonstrates exocytosis, which is how these large molecules exit the cell. In my illustration, you can see the phospholipid bilayer part, and this is how these large molecules exit the cell. An example of this is when cells secretes proteins like enzymes.